I think mass spectrometry is not as prevalent in academic settings because next generation sequencing has been so powerful. And while next generation sequencing has opened up a lot of avenues for us, there are certain biological questions that we can't ask by sequencing. Right? We can't ask which specific proteins are present. The mRNAs might give us a hint at which proteins are present, but just because an mRNA there doesn't mean the protein for that transcript is being made. Also, one of the things we can't get from DNA sequencing is that how proteins are modified. Right? Do they have little chemical additions to them that alter their activity, alter who they interact with, and alter their function? Right? You can't get that from sequencing, so we have to use a mass spectrometer to actually detect those changes in mass and those changes in chemical composition. And that's what this mass spectrometer is particularly good for. In fact, I can only think of a couple labs that are mass spectrometry-based proteomics labs that are mostly emphasizing immunology. And I think historically that's just because the sensitivity of mass spectrometry hasn't been great. But in the last couple of years, it's really gotten to a point where we can do a lot more and we can ask a lot of different questions that weren't able to ask in the past. And immunologists especially are the types of scientists where they care about the mRNA levels and how the mRNA levels translate to protein levels. And those don't always match, especially in a lot of immune system models that we have. And so it's really important that we can do this. So usually with like flow cytometry, which is really big here and very important in immunology, they typically look at up to like 18 proteins per cell, where we can measure 8,000, 9,000 at a time. Not on the single cell level yet, but we can get a much more holistic picture of what's happening. Right now we're in my proteomics facility, the mass spectrometry room. And so we have several pieces of equipment in here that help us accomplish the goal of trying to understand how cells think and make decisions. You'll notice this kind of sound in the background, and what that is that the rough pumps and the turbo pumps that are constantly lowering the pressure of the mass spectrometer. So the mass spectrometer, what we do is we move analytes around, we move ions around in the gas phase. And because they're moving around in the gas phase, we want to have as little gas that our analytes can bump into until we're ready for them to do that. Okay, so first what we'll do is we'll put our samples that we prepared in the wet lab in an auto sampler here. And so what this auto sampler does is we can basically prepare our samples and then put them in these particular HPLC vials, right? Which a needle under here will then dip in here and pull up some sample volume. And then the sample volume will be injected into this liquid chromatography pump, so this nano liquid chromatography setup. And what it is it's basically just two pumps that are pumping very small amounts of liquid where we can combine them together, inject our sample, and then have them go either to the mass spectrometer, or if we need to wash the sample, we can have it go to the wash waste. And basically what this allows us to do is set up a handful of samples at a time so we don't have to be here for each injections, so that it'll automatically run this sample and then wash the column, and then move over to this sample, run it, and then wash the column, and we can just do that indefinitely. One of the things that my lab is particularly good at is improving the sensitivity. That's somewhat to do with the mass spectrometry itself, but it's a lot more to do with the upfront sample preparation that we've made a lot of innovations for. And the reason that's really important is because if you're asking for a specific type of T cell from patient blood, or a certain type of T cell from mice, for example, right? Typically, the amount of protein material that we would need to analyze in kind of classical experiments has been too high for most immunological questions. So with our improvements in sensitivity, both in our laboratory and through the instrumentation advances, we can actually ask these immunological questions because our sensitivity is better, and we can access more and different types of samples that previously weren't possible. So we do chromatic graphic separations on multiple scales, and what that means is that we have a pool of analytes, and let's say there's 1 million things in that sample, and we want to see as many of them as we can. Per unit time, we can only look at, let's say, 100,000, right? So what we want to do is we basically take those 1 million molecules, or those 1 million analytes, and we separate them based on their chemistry. And when we separate them based on their chemistry, we can bin them into things that are more chemically similar, but they're less complex in general, right? So one bin might have the 100,000 things that we can get to in a unit time. And so if we make 10 fractions or 10 bins with the 100,000 different chemically distinct species, then we can go deeper and deeper into our sample. And so we do chromatography to separate these analytes on multiple scales. So one scale that we do is on kind of our high flow scale. And so you can see this is a chromatographic column where we'll pump our analytes through here, and they'll basically interact with the hydrophobic resin that's on the inside of this. And depending on their type of chemistry, they might interact more and slow down, they might speed up and go through, but they'll separate at different times. But basically what I want you to see is the size of this column, okay? So this is doing kind of a large scale, what we would call large scale liquid chromatography separation, okay? And then once we get these particular fractions, which again, we can collect in plates like this where each well will have a different set of analytes that were previously all together, but now they're chemically separated. We can then go in and analyze all the compounds that are in a particular fraction. Okay, so when we run something like this, we run at the hundreds of microliters per minute. So that's basically like how much of the solvents are we pumping through there, okay? And while this is really good for separating these things, that volume, that amount of liquid doesn't necessarily translate well to going into the mass spectrometer. We need as little amount of solvent going into the mass spectrometer as possible, because what we're doing is we're getting our analytes from being in solution, so being dissolved in liquid, to desolvating that, getting rid of the water, and then having it in the gas phase, right? So a complete phase transition. And so when we do that, so if you think about that column that we had that we were separating here, the column that we use to separate online into the mass spectrometer is this here, right? So this is a much, much smaller separation column that we're doing. And we call this online chromatography. And what this allows us to do is take that fraction that still has a lot of material in it, has a lot of different chemical species in it, and they will again separate differently here, and they'll come out a few things at a time. And as they electrospray into the mass spectrometer, we can apply a really high voltage to the liquid that then allows the very small droplets to desolvate and then be pulled, the gas phase analyte will be pulled into the mass spectrometer. So the way we make these emitter columns is that we actually don't make them ourselves. We try to purchase them. This company's gone out of business. But what it is basically they take this capillary and then they use a laser and then shoot a laser in the middle here and then pull it really fast. And that gives it this like really sharp tip here. Okay? And then what's also nice about these is that there's a frit in the end here. So when we try to pack our C18 material, which is these little white beads, right? They don't flow out the end here, right? So the little frit catches them, and then we can pack our column with increasing amounts of this stuff. So basically this is like a big zoom in of this emitter tip, that really sharp capillary I was showing you. So here the opening is only about 10 microns, right? And what we do is we pack these C18 particles, which is hydrophobic. We can pack these C18 particles into this tip so that our peptides will then interact with these and slow down based on their hydrophobicity. And that's how we separate them. That's how we separate our peptides into the gas phase. And then what happens here is we apply the voltage, and then we get an electrospray here where our peptides will go into the mass spectrometer. So these are capillaries that we make into columns. We make into these separation columns that go directly into the mass spectrometer. And so we make these in-house because we find that we get kind of the best separation resolution and the best performance through these that we can't find in commercial products. Right, so what we're looking at here is, so we pack this capillary here with C18 material, this hydrophobic material, right? And then what happens is this packs up and analytes come through here, and then we try to electrospray them through this tip here. So this gives us this really sharp tip that will cause the the droplets that contain our analytes to get really, really small. And then as we apply a voltage, those droplets will desolvate, and then we'll get our analytes into the gas phase. So what we do is we use this camera situation we have here. We have two cameras, one on the top and one on the side, try to point our emitter tip, which emits our ions, our emitter tip as close to that orifice as possible. So what we can do is we can basically eyeball this, and we can move this in 3 dimensions to try to angle it so that it's pointing not directly into the mass spectrometer, because that seems to not work quite as well. We actually kind of angle it so it's not pointing directly in there. And then also we don't want to get it too close, because this is highly charged. There's a really strong voltage that we're applying to the liquid coming out of here. And so if it gets too close to that metal, it'll arc, and then we'll burn the tip, and we don't want to do that. So you can buy components that are kind of plug and play, and I think that works really well for a lot of applications. With our applications, we need such exquisite sensitivity that it's not something we can get from plug and play type pieces of equipment. So that's kind of why we make these things and do all these small tweaks, so that we can actually get to the level of sensitivity, so we can ask these new immunological questions that haven't been asked before. So this is a schematic of the instrument that we have. Okay, so here's where that electrospray ion source is. So that's that little needle I was showing you before, that little emitter tip. And the column would be coming through here, and our HPLC would be pumping our liquid through here, okay? And then we have this high capacity transfer tube, which is basically the only orifice going into the mass spectrometer. And it's high capacity because it's a bigger hole than it used to be. And then it's heated up, so we, again, desolvate. And then our ions, what we can do is we can pass them through here, which focuses in this ion funnel. It focuses our ions. And if they're charged, we can apply electromagnetic voltages to manipulate them. So we have this bent flatipole here. So if it's a neutral ion, it can't be manipulated with electrical fields, so it'll just hit the walls. But if it's charged, we can move it through here. And then this is our first mass spectrometer. And what this allows us to do is filter for things that we want or don't want. So if we want to look at a whole big range of peptides or a single peptide, we can tell this quadrupole to either let everybody through or to let single analytes at a time. And then we can do all sorts of things. This is kind of like the Swiss Army knife of mass spectrometers. So we have another type of mass spectrometer here called an ion trap, and it's very fast and very sensitive, and we can do a lot of gas phase chemistry. And our lab in particular does a lot of specific types of gas phase chemistry on these peptides. And so we really needed this portion here. And then they have another mass spectrometer called an Orbitrap mass spectrometer. So this is not quite as fast or as sensitive, but the resolution is really high. So in a mass spectrometer, in the Orbitrap mass spectrometer, we can tell the difference between a C13, which goes from C12 to C13, or a nitrogen 14 to nitrogen 15. That seems like it's just one additional neutron, but we can actually measure the energy in the nucleus that's keeping the nitrogen or the carbon isotope from each other. So typically the way people look at peptides or sequence a peptide is they move it to this collision cell here, and they slam it into a gas molecule. And so we control the pressure of gas of nitrogen in this cell here. And we basically take our peptide, slam it into a gas molecule, it'll break into pieces, and then we can move those pieces into the Orbitrap to analyze their mass and sequence our peptide. One of the things we do in my lab is we study O-linked glycopeptides, so sugars that are added to proteins and peptides to modulate protein function. And with that, you can't collide them into gas molecules, because what happens is the energy, the bond between the amino acid that's glycosylated and the glycosylation itself is so labile, it's such a weak energetic bond, that it breaks off, and we get no information about what amino acid was modified. And it's really important for us to understand which exact amino acid is modified, so that we can go and do functional studies where we can mutate that residue or monitor it in a healthy control or a disease state, right? We need to know exactly where to look for this modification. So what this ion trap allows us to do is do a reaction called electron transfer dissociation. So instead of colliding our glycopeptide into a gas molecule, we have it react with a very specific negatively charged radical anion, and the electron is tunneled to the glycopeptide, which then does a chemical rearrangement in the gas phase, so we can break apart and sequence our peptide without losing the glycosylation. Right, so it allows us to do gas phase chemistry so that we can maintain the glycosylated site in our analyses. And so what we do is we try to combine a little bit of everything and use the strengths and weaknesses when they can help us. You know, our glycopeptides are not very common, so we spend a lot of time doing the really efficient fragmentation and doing it fast. And then if we see a glycopeptide, if we see a diagnostic ion for a glycopeptide, we then take the time to go into the ion trap and do the electron transfer dissociation, because it's not quite as efficient and it's not quite as fast, but it's really important for nailing down exactly where that glycosylation site was. Okay, so I'll show you what a glycopeptide run looks like. So here what we're looking at is we're looking at the total ion current that is coming into the mass spectrometer, right? So what that means is that each one of these peaks is typically a peptide or two of particular abundance, right? And every time there's a black line, which you can't see right now, that means the mass spectrometer is sequencing a peptide. So if we zoom in here, you can actually see that it's not black. What it's doing is it's doing a scan, and then it takes 2 seconds and looks at as many peptides as it can, and then it takes another scan, and then it looks at as many peptides as it can. So if we zoom out, you can see that this instrument's constantly running.
So how long does this run?
So this run is 110 minutes, right? So there's a gradient that we're separating the peptides on here with increasing organic solvent. So more hydrophobic things will come out here at the end, okay? And what this is a glycopeptide enrichment from embryonic stem cells, okay? And so I can't by eye just tell where the glycopeptides are, so what we can do is we actually have a way that we can extract ions that we want to look for. Okay, so what this is doing is it's saying we're looking for every 204 ion that we can see in this particular run, right? So each peak here now is a 204 ion. And the reason that's important is because the 204 ion is the mass of the glycnac, the glycosylation that we're looking for, having been knocked off the peptide, the glycopeptide. So what we can do is we can actually tell that these are glycopeptides. So if I click here and I look down here, what I can see is I can see fragmentation of our glycopeptide, okay? And we'll be able to determine the sequence of the peptide from these changes. And then here's where it's beautiful. This is the 204 ion. This is our glycnac peak. We call it our hexnac peak, but we know for certain probably it's a glycnac. And then this is our little hexnac fingerprint here. The 204, the 186, the 168, the 138, and the 1. This is a signature of a glycopeptide. So we now know for certain this is a glycopeptide, okay? And so what we will tell the instrument to do is to take note that this is a glycopeptide. And so 843 is the sample, and now it does electron transfer dissociation on it. So now we see a different type of spectrum that'll help us identify the site more specifically. Okay, so this is a sample that wasn't enriched for glycopeptides, okay? And you can see we still see a lot of ion current. We still see the instrument's working hard. If we ask it to look for evidence of glycopeptides, we don't see a lot of peaks, right? There's always some abundant glycopeptides in your sample, but you can see there's these valleys here that are just, there's nothing going on, okay? But if we turn to a sample in which we've taken steps to actually enrich for these glycopeptides, to enrich out of the milieu of all these unmodified peptides, then what we can do is we can then say, well, how much of our ion current has a diagnostic ion for a glycopeptide? Okay, and so when we look for glycosylation ions in a sample that we enriched for the glycopeptides, in contrast to seeing very little in our diagnostic peak, now we see a lot of signal, right? So this is how we know we've enriched for glycopeptides really well, okay? And so this is allowing us to identify these glycopeptides for the first time in a throughput that hasn't been available before. I feel so warm inside when I look at these is because I know I want to go in and look at these by hand. And back in the day, what you'd have to do is you basically find your diagnostic ion, right? So here's a peak for our hexnac, our glycnac, and then basically I would go in, I would make sure it has the diagnostic ions to tell me it's a glycopeptide. And then I would look at this by hand and say, oh, I can tell the mass difference between here and between here, right? And I can sequence this peptide by myself. But that was in the days when we were identifying 100 of these. Now we're identifying several thousands of these. And so we turn a lot more to automated software to help us kind of initially find things. And then for ones that are particularly important or ones that we think might be important for immunological function or disease states, those are the ones we'll go in and look at by hand to make sure that the software is calling them correctly. So we're asking new biological questions and we're approaching immunological challenges from a new perspective. But as far as the instrumentation goes, I'm standing on the shoulders of giants.